Hello friends, welcome once again in another video lecture of Easy Life Science. Here I will be talking about some important questions, their answers with detailed explanation which are asked for a SET exam, subject Life Science part 14. If you like my video, please subscribe my channel Easy Life Science, share it to your friends and don't forget to press the bell icon so you will get the notification of my newly uploaded video. So let's start. Which of the following is a post-zygotic isolating mechanism in a speciation? Four options are there: behavioral isolation, seasonal isolation, fertilization failure, and hybrid sterility. The post-zygotic isolation occurs after the members of two different species have mated and produced a zygote. Offsprings of such mating are known as hybrids. Hybrids are unable to reproduce themselves, so the production of hybrid is not considered a successful mating. We know the reproductive isolation is the mechanism that keeps the species from mating with other species. Simply we can say that the goat cannot mate with a dog or dog cannot mate with a cat. If they mate with each other, they cannot form a fertile offsprings. There are two mechanisms of speciations. One of them is a post-zygotic and another one is a pre-zygotic. The pre-zygotic isolation prevents the fertilization of eggs, while post-zygotic isolation prevents the formation of fertile offspring. Pre-zygotic isolation mechanism includes habitat isolation, mating seasons, mechanical isolation, gamete isolation, and behavioral isolation. Whereas the post-zygotic isolation mechanism includes hybrid sterility, hybrid inviability. Pre-zygotic barriers occurs before the fertilization and making it impossible for members of different species to mate. Example of these barriers includes differences in a habitat, mating rituals, whereas the post-zygotic barriers occurs after the fertilization and results in reduced zygote viability or offspring with lower fitness. Some examples of post-zygotic isolations are: one of them is a hybrid sterility. The mules are the hybrid offspring of the horses and the donkeys. They are generally sterile because of chromosomal mismatches between horse and the donkey. This means that they are unable to pass on their genetic material. This is one of the post-zygotic isolation mechanism, that is a hybrid sterility. And in this question is asked on which of the following is the post-zygotic isolating mechanism in a speciation. So the answer is a hybrid sterility. Also, low hybrid viability is also reason for a post-zygotic isolation mechanism. Low hybrid viability means certain species of a frog can form a hybrid tadpoles. However, the hybrids are not viable and die before they can become a sexually mature. Zygote mortality: it is possible for goats and sheep to mate and for fertilization to occur, but the genetic differences result in a zygote that does not develop properly and will die. So, low hybrid sterility, low hybrid viability, and zygote mortality are the post-zygotic isolating mechanisms, whereas behavioral isolation and seasonal isolations are pre-zygotic isolations. So, answer for this question is option D, that is a hybrid sterility. Then next question: a species facing an extremely high risk of extinction in the immediate future is known as. So answer of this is option C, that is endangered species. A species facing an extremely high risk of extinction in the immediate future are known as endangered species, option C. An endangered species is a type of organism that is threatened by the extinction. Species become endangered for two main reasons. One of them is a loss of habitat and a loss of genetic variation. or a species considered to be facing a very high risk of extinction in the wild. For example, the Javan rhinoceros, Amur leopard, and Sundara Iceland tigers, mountain gorilla are some examples of such endangered species. The option A, that is a vulnerable species. A species is vulnerable if its population has declined at least 50% and the cause of decline is known. Habitat loss is the leading known cause of a population decline. A species is also classified as vulnerable if its population has declined at least 30% and cause of decline is not known. Then the option B, that is a rare species. Rare species are those which are uncommon in nature and exist in very low population. Vulnerable species, on the other hand, neither less populated nor uncommon. Then the option D, that is a link species. Link species is defined as the species which shows characteristics form to different species. Example, lovefish is the link species between fishes and amphibians. For this particular question, option C is the answer, that is an endangered species. Here about the pre-zygotic isolation and post-zygotic isolation. The geographic isolation, the physical barriers like rivers, oceans, mountains, these barriers does not allow the mixing of a population. So it is a geographical isolation. Then ecological isolation, a species occur in same area but inhabit to different habitat, so they cannot encounter each other. Temporal isolation, species reproduce in different seasons or at different times of the day. Behavioral isolation, species differs in their mating rituals. For example, different bird songs are different songs, mating colors and dances and the pheromones are the some examples of behavioral isolations. Mechanical isolation, the body structure of the two different species prevent themselves to mating. And gamete fusion, gametes of the different species cannot fuse with egg of female species. So this is about the pre-zygotic isolation. Then the post-zygotic isolation, it appears after the formation of zygote. It may be a hybrid zygote when the two species meet each other and forms a hybrid. Infertility, natural selection and hybrid invariability are some mechanisms of post-zygotic isolation. Here in this image, there are the differences in between the post-zygotic isolation and pre-zygotic isolation. First column, pre-zygotic isolation. This type of isolation where the reproduction occurs, zygote is formed, but in case of post-zygotic isolation, there is a zygote formation, but zygote cannot lead to formation of a matured organism. In a pre-zygotic isolation, mating does not occur, but in a post-zygotic isolation, two different species can mate with each other. Then moving to the next question. In zoo, lions and tigers mate and produce a viable offspring called as a tigons and a ligers. Hybrids were near recorded in Asia. However, where their ranges once overlapped, hybrids were probably not produced because of. This is an example of the speciation. Answer of this question is a behavioral isolation. Option C. The hybrid infertility. Option A. It is a post-zygotic barrier which reduces a hybrid's capacity to mature into a healthy fit adults. Low health of these hybrids prevents gene flow between the species. Thus, the hybrid infertility or inviability acts as a isolating mechanism. Then option B, gametic mortality. Gametic mortality or a physiological isolation is a typical pre-zygotic reproductive isolation mechanism in which gametes die before a zygote can be formed. Usually, sperms are unable to survive in the female reproductive tract of the members of wrong species. This is known as a gametic mortality. Then the behavioral isolation, it is defined as the species are isolated reproductively from other species due to differences in behavioral. Behavioral isolation is the way in which nature prevents interbreeding of species through a behavioral isolation. Example of this, male fireflies use a specific light and this light pattern attracts a female. Then the option D, mechanical isolation. It is a physical incompatibility between reproductive organs of the two organisms. Mechanical isolation is the type of pre-zygotic barrier which no fertilization occurs and thus reproduction does not occur. Mechanical isolation includes any physical barrier that prevents a mating. So for this particular question is asked about the tygones and ligas. And the tygones and ligas does not form a further offsprings. These are the hybrids. So on the basis of a behavioral isolation, they cannot form further offsprings. Then in the next question, the word filial is used for. The word filial is used for a progeny. The meaning of this word is having or assuming the relation with a child or the offspring. So option B is the answer of this question. Then the next question, the basic idea of evolution is. The basic idea of evolution is descent with a modification. Option A is the answer of this question. Descent with modification means that traits are passed down from generation to generation and sometimes undergo a changes or a modifications over time. These changes may be caused by natural selections or even a mutation of a DNA. The term became popular when the Charles Darwin used it in his book, the book named as A Descent of Man. For example, imagine the population experiences many years of a drought in which there are a few plants that the beetles eats. All the beetles have the same chances of survival and reproduction. But because of food restrictions, the beetles in a later generation are smaller than pre-drought generations of the beetles. Option B, that is a spontaneous generation. The idea of a spontaneous theory put forth by Greek philosopher Aristotle. The Aristotle proposed that life can arise from a non-living matter. And the example given by the Aristotle was, dust creates flies, maggots arise from rotting meats, and the bread or a wheat left in a dark corner produces a mice. Means these all non-living things give rise to a living one. So this is about the spontaneous theory put forth by the scientist Aristotle. And this theory disproved by Louis Pasteur. And the option C, a spatial creation. A theory of a spatial creation says that all living organisms on the world were created by a God. God created a first man, that was the Adam. Father Soros was the strong believer of this theory, and this theory is known as the theory of a spatial creation. So, basic idea of evolution is a descent with modification given by the scientist Charles Darwin. Option A is the answer of this question. Then moving to the next question, which of the following was the earliest form with liquid bilayer and can reproduce by a budding? Answer of this is the option B, that is a microspheres. Microspheres are a microscopic firm spherules which form on the cooling of hot saturated solution of proteinites. They were first reported in 1959 by scientist Stanley Fox, who proposed that microspheres might represent the significant early stage in a pre-cellular evolution. Option A, quasarwaves. Quasarwaves are a large collider particles that participates or out in an aqueous medium. Quasarwaves are the first pre-cells which gradually transformed into a living cells. A quasarwave is a spherical aggregation of liquid molecules making up a colloidal inclusions which is held together by hydrophobic forces. Probiotic. These are nothing but the useful lactic acid bacteria which are present in the intestine of the animals or the human beings. So answer of this question is a microspheres. Then the next question, the raw material for evolution is variability of gene or alleles in. The raw material for evolution is variability of genes or alleles in a gene pool. Option C is the answer of this question. Gene pool refers to the combination of all the genes including alleles present in a reproducing population or a species. A large gene pool has extensive genomic diversity and is better able to withstand environmental changes or challenges, or we can simply define we can simply say that a gene pool is a collection of different genes, both expressed or not expressed, present in a population of particular species. For example, frogs in a ponds and frogs in a trees present in a forest. So option C is the answer of this question. Then the next question, kanamycin A, B and C are produced by. The kanamycin is a antibiotic. It is a aminoglycoside antibiotic used to treat different types of bacterial infections. Kanamycin works by binding to the 30S ribosomal subunit, causing misreading of mRNA and leaving the bacteria unable to synthesize proteins which are vital for their growth. So kanamycins inhibit a protein synthesis of the pathogen or a bacteria by binding to a 30th subunit of a ribosome at time of protein synthesis. Kanamycin A, B and C are produced by organism Kanamycetes. Option D is the answer of this question. Then the next question, which of the following is a biopolymer? Option D is the answer of it, polynucleotide. It is a biopolymer or polynucleotide. It is also known as a DNA. Next question, technique used for separation of a protein of identical molecular weight and different charges. Here the question is asked about the differentiation or a separation of proteins on the basis of their charge which having a same molecular weight. Technique used for a separation of a proteins of identical molecular weight and different charge is a isoelectric focusing. The isoelectric focusing is also known as electrofocusing. It is a technique for separating different molecules by differences in their isoelectric point. The main principle of isoelectric focusing is that two different proteins having different isoelectric points migrates in a pH gradients and presence of electric field till total charges of protein get to the zero. First dimension in the 2D gel electrophoresis experiment involves separation of a protein according to their isoelectric point. So answer of this question is option B, isoelectric point. Let's see the other options. Option A, SDS page, sodium dodecyl sulfate polyacrylamide gel electrophoresis. Sodium dodecyl sulfate polyacrylamide gel electrophoresis is the technique used for separation of proteins based on their molecular weight. Principle of SDS page states that charged molecules migrate to the electrode with opposite charge when they are placed in an electric field. SDS is a detergent present in a sample buffer SDS along with some reducing agents functions to break disulfide bonds of the proteins. Option C, the native page. It is a technique that uses non-denatured gels for the separation of protein. Unlike SDS page, no denaturing agent is added in the preparation of gel. As a result, the separation of a protein takes place on the basis of charge and size of protein. In native phase, proteins are separated according to net charge, size, shape of their native structure. And the option D, agarose gel electrophoresis. In agarose gel electrophoresis, macromolecules such as DNA or a protein in a matrix of agarose gel are separated. The protein separated by a charge and or size, and the DNA and RNA fragment by a length. Biomolecules are separated by applying an electric field to move the charged molecule through an agarose matrix. So option B, that is isoelectric focusing, is the technique used for separation of a protein which having a identical molecular weight but different charge. Then the next question, RAPD stands for random amplification of polymorphic DNA. Random amplification of polymorphic DNA. It is a type of polymerase chain reaction, but the segment of a DNA that are amplified are random. So option B is the answer of it. The next question, in flow cytometry, side scattering of light measures the cell granularity and complexity. Option B is the answer of this question. Flow cytometry is the technology that rapidly analyzes single cells or particles as they flow fast single or multiple lasers while suspended in a buffered salt based solution. Each particle is analyzed for visible light scattering and one or multiple fluorescence parameters, or simply we can say that the flow cytometry is a laser based technique used to analyze the chemical and physical characteristics of the particles. The next question, the helix content of the protein can be determined specifically by. Answer of it is the option C, that is a circular dichroism spectrometer or CDS. The CDS uses circularly polarized light. It is used to investigate secondary structure of the protein. Option A, infrared spectrometer. The IR spectroscopy utilizes the concept that molecules tends to absorb specific frequencies of light that are characteristics of corresponding structure of molecule. And the fluorescence spectrophotometer, it is the type of electromagnetic spectroscopy that analyzes fluorescence from a sample. It involves using a light beam, usually UV light, that excited the electron in a molecules of certain compounds and causes them to emit the light. So option C, CDS, circular dichroism spectrometer. By this technique we can determine the helix contents of the proteins. Then the next question, the phenomenon of piezoelectric effect is used in a sonography. Piezoelectric effect is the ability of certain molecule to generate an electric charge in response to applied mechanical stress. This principle is used in a sonography. Then the next question, blue baby syndrome is caused by. The blue baby syndrome is caused by a excess of a nitrate. The use of nitrate contaminated drinking water to the infant causes infant methemoglobinemia. Affected infant develop a peculiar blue-gray skin color. The nitrates can stop the red blood cells from doing their job of carrying oxygen. When RBCs in a baby does not carry oxygen, it can make a baby's skin blue. Methoglobinemia is the disease which is caused by use of excess nitrate. It is also known as a blue baby syndrome. Then the next question, which of the following pyramids is always upright? Read this question carefully. Here the question is asked, always upright means like this: pyramid of energy, pyramid of biomass, pyramid of number, and none of above. And we know that there are the three types of ecological pyramids, includes pyramid of numbers, biomass, and energy. Pyramid of energy is always upright, can be never inverted because when the energy flows from a particular tropic level to the next tropic level, some energy is always lost as a heat in each step. Here in this image, here three pyramids are there: pyramid of energy, pyramid of biomass in a ocean, and pyramid of number. And this pyramid of energy, is always upright. The pyramid of number is not inverted or upright. For example, pyramid of number is inverted in case of parasitic food chain and forest ecosystem. Pyramid of number represents the number of organisms at each tropic level. The pyramid of number is usually upright except for some situations like detritus food chain, where many organisms feed on one dead plant or animal. In a tree ecosystem, the number is inverted because the number or a biomass at the producer level is minimum and increases at the each tropical level, and is similar in the case of the pyramid of sea ecosystem. So, pyramid of number is not always upright. Then the pyramid of a biomass is a representation of a total living biomass or organic matter present at different tropic levels in an ecosystem. Pyramid of biomass is usually depicted as an upright but not always, because the biomass of aquatic ecosystem is inverted. Here the biomass of primary producer is much less than the zooplanktons. Which is less than the small fish and the big fish having a maximum biomass. So, answer for this question: which of the following pyramids is always upright? Answer of it is a pyramid of energy. The next question: myelinated nerve fibers differs from non-myelinated nerve fibers in. Myelinated nerve fibers occurs in the white matters of the brain, spinal cord, and in the central and cranial nervous system. Unmyelinated nerve fibers occurs in the autonomous nervous system. Myelinated nerve fibers contain a myelin sheath around the nerve fiber, whereas in unmyelinated nerve fibers do not have any myelin sheath around a nerve fiber. Myelinated nerve fibers are white in color, whereas unmyelinated nerve fibers are grey in color. Myelinated nerve fibers consist of modes of Ranvier, whereas unmyelinated nerve fiber does not consist of nodes of Ranvier. In myelinated nerve fibers, transmission occurs only through nodes of Ranvier. The speed of transmission of nerve impulses is high. The speed of transmission of the nerve impulses in unmyelinated nerve fiber is very low because they do not contain a myelin sheath. The myelinated nerve fibers include most of peripheral nerves, whereas unmyelinated nerve fiber includes small axon neurons in the CNS. Long axon nerve fibers are myelinated, whereas short axon nerve fibers are unmyelinated. Myelin sheath in a myelinated nerve fiber prevent a loss of impulse during a conduction, whereas the unmyelinated nerve fiber can lose the nerve impulses during a conduction. Question: myelinated nerve fiber differ from a non-myelinated nerve fiber in showing a saltatory conduction of impulses. Option C is the answer of this question. Then the next question, a taxonomic system which weigh only a phenotypic characteristics is known as. It is known as a phonetics. A taxonomic system which weighs only a phenotypic characteristics is called as a phonetics. Phonetics is an attempt to classify the organisms based on overall similarities, usually in morphology or other observable traits, regardless of their phylogeny or evolutionary relation. So option B is the answer of this question. The read other options like phylogeny. The phylogeny, in this phylogeny, classification of organism is based on evolutionary history and a common ancestry of the organism. It gives us the underlying cause of the differences between genetic diversity of the organism. Generally, the connections between all the groups of organisms and the species with ancestor and descendants relationships are understood. These relationships are determined by phylogenetic inference that focus on observable heritable traits such as DNA sequence, protein amino acid sequence. The results of such analysis is known as a phylogenetic tree. Option C, cladistic. It is an approach to biological classification in which organisms are categorized into groups, that is clades, based on a hypothesis of most recent common ancestry. So option B is the answer of this question. Reading the next question, phenology is the study of, it is a study of periodic plant and animal life cycle. Phenology is the study of periodic events in a biological life cycles and how these are influenced by seasonal and interannual variations in the climates as well as habitat factors. So option C is the answer of this question. Next question, which of the following causes a lockjaw disease? Lockjaw disease, the lumpy jaw disease, is also known as a tetanus. This disease is caused by a Clostridium tetani. It is an anaerobic spore forming organism produces a toxin. When these bacteria enters the body, they produce a toxin that causes painful muscle contractions. It often causes a person's neck and jaw muscle to lock, making it hard to open the mouth and the mouth gets swallowed. So causative agent for the lockjaw disease is Clostridium tetani. Read the other options like Bacillus anthracis. It is a spore forming gram positive bacteria, also produces a toxin, and this produces a toxin named as the anthrax. And this organism, Bacillus anthracis, spores of these organisms were used for a bio war, causes a disease that is anthrax. Option C, Corynebacterium diphtheria. This causes a disease that is a diphtheria. Mycobacterium species, Mycobacterium lepri causes a leprosy. Mycobacterium tuberculosis causes a TB. So option A is the answer of this question. Then the next question, when a population enters a new niche or a habitat within a range of a parent species, this form of speciation is known as a parapatric. Here the population does not get separated, it has connected with a original one, so it is the parapatric. If it has a different niche or a different habitat, then we can say that such speciation will be the allopatry. So option A is the answer of this question. Thank you friends, thank you for watching my video. If you like my videos, subscribe my channel Easy Life Science and share my video to your friends, and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. Thank you once again.